Hi students, welcome to SPS University. Follow us on social media. This is Suresh, Senior Biology Faculty. So we are discussing about the molecular basis of inheritance, in which we discussed about the DNA, RNA. So now we are going to discuss about the regulation of gene expression. What is this regulation of gene expression? So when we come to regulation of gene expression, so it is carried out by, that is, Francis Jacques Monod of Pasteur Institute, and they have done research on E. coli. So E. coli will synthesize enzyme if and when it is appropriate to do so. And based on this, they proposed the operon model. And they awarded Nobel Prize in 1965 for this operon model. So what is this model? What it says? So what this operon model says about this, that is, when you come to operon model, an operon is an transcriptional unit that includes structural genes, a promoter, and operator. That is, according to Jacob and Monod, an operon is a transcriptional unit that has structural gene, promoter, and operator. Okay. So here if you see the diagram very carefully, so this is regulatory gene I, promoter gene P, operator gene O, and structural genes, that is, three structural genes are present. One is LAC-Z, LAC-Y, LAC-A. What LAC-Z will do is it will synthesize an enzyme called beta-galactosidase. LAC-Y, it will secrete an enzyme that is permease. And LAC-A will secrete an enzyme called transacetylase. So on transcription, the structural genes on transcription and translation, they will produce enzymes which will play an important role in the metabolism of the lactose sugar. So that is, in the presence of lactose sugar, these three enzymes are synthesized. In the absence of this lactose sugar, these three enzymes are not synthesized. How it is possible? What is this? So according to, when you come to the structural gene, so a gene that actually controls the protein production. So here the protein means nothing but the enzymes. Enzymes are made up of proteins. So in structural gene, there are three genes, LAC-Z, Y, A. LAC-Z will produce beta-galactosidase. LAC-Y will code for a protein permease. LAC-A will code for an enzyme transacetylase. So these are three structural genes. Come to operator gene. What operator will do? It is a part of operon that controls the activity of the structural genes. How it will control? How it is possible? So to this operator gene, a repressor protein is attached. So repressor is a protein to which attached. When it is attached, it will stop transcription. That means it will stop the transcription of structural genes. So when the structural genes transcription is stopped, so there is no production of the enzymes which are produced by structural genes. So no transcription, no enzymes. So this is how operator gene will control the transcription of the structural genes. Next come to operator. So operator in promoter gene, that is promoter gene, it is a nucleotide sequence to which the enzyme RNA polymerase will attach. So RNA polymerase is an enzyme which initiates transcription. So promoter is a place where the enzyme binds, that is the RNA polymerase binds to the promoter. It is the place where the RNA polymerase binds to the promoter. What is regulator? A regulator is, it produces a protein. So that protein name is repressor. For blocking RNA polymerase. That is, this protein which is synthesized from regulator gene, it attaches to operator. That is, regulator gene will form a protein, it will code for a protein, that is repressor, which attaches to operator. So operator, promoter, regulator, structure has each and has a very specific role. So here whatever this is, that is whatever the operator, that is the regulator, the structural genes which will form proteins, that is they will form proteins by transcription. On transcription RNA is formed, on translation protein is formed. And these proteins act like enzymes. So we'll discuss about, now we are going to discuss about an inducible operon. So in this operon, it is related to the metabolism of lactose. That is, in the absence of lactose, what will happen? That means it will be in switch off state. What is this switch off state? What will happen during this process? During this process, when lactose is not present, regulator gene will on transcription and translation. So on transcription as well as on translation, it will form a repressor protein. So and this repressor protein binds to operator. So when it binds to operator, whatever the RNA polymerase enzyme is present, it cannot cross this operator. That means it cannot reach to the structural genes. It will stop here itself. When it stops here itself, there is no transcription. When there is no transcription, obviously there is no translation. So no translation means no protein coding. So no protein means no enzymes are formed, obviously. That means when lactose is not present, there is no need of synthesizing enzymes. That's the reason why this is how it is switched off. Here when you come to this, okay, here if you see this, that is in the absence of lactose, there is no need of producing any of the structural enzymes, that is permease, the beta-galactosidase, the permease and transacetylase. No need of these enzymes. That means there is no lactose metabolism at all. It is just like the requirement of our light in our house. When we need, we switch on. When we don't need, we switch off. The same way, Genes are like switch on and switch off mode. Whenever needed, they will become active. Whenever they don't need, they don't show any transcription. Okay. So in this condition, the structural genes are switched off. When structural genes are switched off, so there is no synthesis of enzymes. Okay. This is when it is in switched off condition. Okay. So here what happens is, here how the structural genes are switched off? By blocking RNA polymerase. So RNA polymerase is not allowed to move beyond this point. This is how the switch off. Come to switch on condition. What happens here? In switch on condition, that is lactose is present. So this lactose at this condition, it is called inducer. When lactose is present, this lactose binds to the repressor. So when it binds to repressor, the repressor becomes inactive. When the repressor becomes inactive, the RNA polymerase can move towards structural genes, so which leads to transcription. So here there is transcription, and after transcription, that is after formation of the mRNA, next it takes place, that is translation. After translation, these three enzymes are formed: beta-galactosidase, permease, transcyclase, which play an active role in lactose metabolism. That is, in the presence of lactose, the repressor is inactivated, so that the RNA polymerase can proceed towards the structural genes. And these enzymes are needed for lactose metabolism. So when there is the requirement of lactose metabolism, these enzymes are synthesized. So these enzymes are synthesized. Okay. So here the lactose binds to the repressor, so that the repressor becomes inactive. So now it is in switch on condition. So the lactoperon is now on a switch on condition. Okay. So here, what is the purpose to make it inactive? The purpose is it cannot bind to operator. If it binds to operator, again the process will stop. So to make it not binding to operator, it is made inactive, so that the enzymes, that is structural genes, will undergo normal transcription, translation, which will form enzymes. So here you can see the comparative study of, that is in the absence of inducer. Inducer means here lactose, that is switch off. In the presence of inducer, that is switch on. So both. In the presence of lactose, in the absence of lactose. Here what happens? The repressor binds to operator. So no transcription here. In the presence of lactose, that is the lactose binds to repressor where it becomes inactive. So there is a transcription which leads to formation of these three enzymes. Okay. So here there is a control on as negative as well as positive. Okay. So when there is negative, that is repressor will be active, transcription will be off, and repressor will be active, which will inhibit transcription. Okay. So obviously the process will stop. So this is how the genes are regulated at different levels, not only at transcription level, even at translation level, even at pre-translation stages. There are regulation of genes at different points. And gene is like a switch on and switch off mode. So follow us on social media, SPS VERSITY. Okay. Okay. So we discussed about that is the regulation of gene. So now we are going to discuss about Human Genome Project. What is meant by genome? A genome is a complete set of DNA where it includes all the genes. That is, for example, if we take humans, the diploid chromosome number of humans is 46 chromosomes, or we can write it as 23 pairs. In this 23 pairs, 22 are autosomes and one pair is 6 chromosomes. That is, if it is female, there are 2 X chromosomes. If it is male, 1 X and 1 Y chromosome. So that is, total if you see this, that is, hence a complete set of human genome. So consists of, that is, you can see that is 23 pairs of chromosomes. Okay. So here in this Human Genome Project, the main aim of this is to find out all the genes present in the genome, their sequence, and to find out the sequence of entire genome. Okay. To find out the sequence of this entire genome, so this is a big project where roughly it will take about 13-year project, where it is initiated by U.S. Department of Energy and National Institute of Health. Later on, Wellcome Trust, next partner is Wellcome Trust of UK. Later on, many other countries has joined: Japan, France, India, Germany, China also. It is started in 1990 and is completed in 2003. Okay. So why it is called mega project? What is the reason for this to call it as a mega project? That is, human genome consists of 3 into 10 to the power of 9 base pairs. Okay. To find out the sequence, that is per base pair, it takes nearly $3. It means roughly the approximate cost of this project is $9 billion. So that's why it is called mega project. And for this project, lot of money is involved. That is, roughly it will cost about $9 billion. And if you start writing the sequence in books, it takes roughly about 3,300 books. That is, if each book has 1,000 pages per book and there are 1,000 letters per page, total it will be a 3,300 books. That is, to write the entire sequence of human genome. In this Human Genome Project, along with the branches of biology like genetics, biochemistry, along with this, there is another branch of biology has developed, that is bioinformatics, where it involves the other branches of sciences like engineering, computer science, chemistry, biochemistry, biology, statistics, and mathematics. So this bioinformatics, the information is stored in form of databases. The information is stored in the form of database. Okay, that is, it is stored in, that is, in systems. Okay, so bioinformatics deals about the storing of data. Okay, analyzing the data. So what are the goals of Human Genome Project? The goals are, that is, to identify all the genes and determine the sequence of all 3 billion base pairs which are present in human DNA. Store this in the form of database, that is, in systems to store the data in computers, and analyze the data which is stored and use this data for technologies in other sectors like medical industry, and address many ethical, legal, and social issues. Okay, so which may arise from the project. So these are the goals of Human Genome Project. Okay. How to achieve these goals? What is the method to follow? So here, to sequence the entire genome, there are two methods. One is expressor sequence tags and sequence annotation. Expressor sequence tags, it sequences only the functional DNA, that is, which is active DNA. Whereas when you come to sequence annotation, it will sequence the entire genome. Entire genome means it will sequence both functional as well as non-functional. So in this Human Genome Project, sequence annotation is selected. That is, in Human Genome Project, the entire genome of human DNA is sequenced. There are other methods also, techniques like polymerase chain reaction, restricted fragment length polymorphism, RFLPS, and PCR. Okay. And there is another method called shotgun technique. Okay. So this shotgun technique, a DNA is collected, many copies of DNA are made, and these copies are broken down into many pieces. Okay. And these sequences are arranged and the complete genome is assembled. Okay. So like this, the human genome is collected from, that is, human genome is sampled, is collected from a source, and it is sequenced by using the technique that is sequence annotation. So in this sequence annotation, the entire genome is sequenced. When the entire human genome is sequenced, what is the output of this? So the output of this is human genome contains total 3164.7 million base nucleotides. Okay. And the average gene consists of 3000 bases. And the lossless gene is dystrophin, which has 2.4 million bases. And the total number of genes is 30,000, which is much lower what we expected, that is 80,000 to 1,40,000 genes. So here, previously we used to think that is there are 80,000 to 1,40,000 genes. Okay. But now it has come to estimate that is there are only 30,000 genes. Okay. So here there are 99, that is, if you take this, in all the human beings, that is 99.9% nucleotide bases are seen, but only a difference will be there 0.1. Okay. So here, and the next important thing is, that is, the function of 50% of DNA is unknown, and less than 2% of genes code for proteins. That means 2% is only functional. And whatever the genome is present, it consists of repeated sequences. It is made up of large number of repeated sequences. That is, it is called repetitive DNA. So most of the DNA which is present, it is repetitive DNA, where it has the repetitive sequences of DNA. Okay. And these repetitive DNA sequences does not have any coding function. Okay. But they reflect on the chromosome structure, dynamics, and evolution. And in these chromosomes, the chromosome 1 has most genes, that is 2968, and Y has the fewest genes, that is 231. Okay. And in this, there are about 1.4 million locations where there are single base DNA differences, which is called SNP, single nucleotide polymorphism, which occur in humans. Okay. So what we will do with this? What are the applications? What are the challenges which are chased by this human genome? That is, for better understanding of biological systems and a new approach to biological research, understanding genetic diseases and curing of genetic disorders, tracing evolutionary links. A lot of evolutionary links are still unknown. They can be traced by this Human Genome Project. So Human Genome Project plays an important role not only in the field of medicine as well as in the field of evolution also. Okay. And it helps us to understand the various biological systems which are surrounded by us. So that's how Human Genome Project will help us. So follow us on SPSVERSITY on social media. Okay. So now we discuss about the Human Genome Project, which is a mega project. And next we are going to discuss about DNA fingerprinting. What is this DNA fingerprinting? So what is this DNA fingerprinting? So it is a technique used by scientists to distinguish between individuals of same species. That is, by using only samples of their DNA. Okay. By taking only the samples of DNA, okay, we can distinguish between individuals of the same species. For example, if we take human beings we can, which belong to the same clade or not, we can identify. Okay. So here, before going into the human DNA fingerprinting, we just have a recap what we studied. Okay. So here if we take each and every cell in our body has DNA. Okay. So 99.9% of DNA into humans is same. That means if we take whatever the human being, the 99 point, only 0.1 makes the difference. Okay. See, the 0.1% is making such a large difference. Okay. And where is this 0.1% difference? So large quantity of repetitive DNA exists in human genome, so which is referred as satellite DNA. That means in our DNA there is a satellite DNA which is having the mini satellite and micro satellite. So what is this mini and micro satellite? Okay. So when you take satellite DNA, in satellite DNA there are again mini and micro satellite. So mini satellite is nothing but a short sequence of repetitive DNA. Show greater variation from one person to the next. Okay. And these mini satellites, as they are present in repeated DNA, do not code for proteins. Okay. But appear throughout the genome of hundreds of times, with many repeated copies lying next to each other. Okay. That is, the variation is seen in number of repeated units. Okay. So this variation which is seen in large, that is, it is called variable number of tandem repeats. So what is this variable number of tandem repeats? What are these V and TRs? So V and TR are variable number of tandem repeats which form the basis for the DNA fingerprinting. And this DNA fingerprinting was invented by Professor Alex Jeffries. So this technique says that simultaneously many satellites in the genome will produce a pattern unique to each and every one. So the genome which is produced, this pattern is unique to each and every individual. So it is referred as DNA fingerprint. Okay. And this DNA fingerprint is unique to each and every one. It is not like same to everyone. So how to find out the sequence of VNTR? By using probes. So these probes, what are these probes? These are single standard radioactive short sequences of DNA which are complementary to VNTR. That is, to find out the sequence of VNTR, we use probes which are, that is, we use probes which are complementary to, we use probes which are complementary to the VNTR. Okay. So here these probes are radioactive short sequences of DNA which are used to find out the sequence of VNTR. Okay. So here when you come to this DNA fingerprinting technique, in this there are many methods are used. Like one is gel electrophoresis. What is this gel electrophoresis? It is a technique used to separate charged molecules. That is, by using this gel electrophoresis, we can separate DNA, RNA and proteins which has charge according to their size. And these charged molecules will move through the gel when electricity is passed. Okay. And these molecules will move towards opposite charge. So here smaller molecules will migrate faster than larger fragments. That is, gel electrophoresis is used to separate charged molecules like DNA fragments, smaller fragments, larger fragments. They can be separated. Here whatever the gel is used, that is agarose gel, which can be used to separate the DNA fragments, which can be used to separate them. Okay. So here this electrophoresis is used to separate DNA fragments according to their length. According to their length means all small fragments will come to one end, larger fragments will be other end. So here DNA is a negatively charged one, hence it always migrates towards positively charged. And during this electrophoresis, fragments are arranged in order of size, which appear like bands. So which appear like bands means like this. Smaller fragments, larger fragments, all are separated according to size. A DNA marker with fragments known lengths is taken from samples. So by comparing the bands of the DNA samples from the DNA marker, we can find out the length of DNA fragments in samples. So electrophoresis is meant to separate DNA fragments of various lengths. So here you can see the gel electrophoresis apparatus. So this is the gel electrophoresis apparatus. These are wells in which the DNA fragments are deposited. On both the sides of the DNA fragment, it is connected to electrodes. So this is positive, this is negative. Okay. And in this there is a gel, that is agarose gel. So when electricity is passed, we can see the DNA is separated like bands. Here you can see in this picture, the DNA is separated like bands. So by using ultraviolet rays and ethidium bromide, you can see the bands. Okay. So according to their size, we can see different bands. And if we take the audiograph, or that is audiography of this, we can see like this, a picture like this, which are separated according to their bands. So what are the steps of DNA fingerprinting? That is, the first step is extraction of DNA. The second step, The extracted DNA is digested, that is, it is made into fragments by using enzymes called restriction endonucleases. Whatever the restriction endonuclease is separated, that is, the separation of fragments by gel electrophoresis. And later on, these separated DNA fragments are transferred to nitrocellulose paper. And later on, they are hybridized with probe. And last, by using X-ray, they take an autoradiography. Okay, where we can find out PNTRs. So from where we'll get this DNA? What is the source of DNA? For example, if any issue occurs, any crime happens. So from the evidences, that is, from evidences, the DNA is collected. So DNA means from hair. Evidences like hair, fingerprint, blood samples, like this. Small tissues, saliva. These all are evidences collected from the crime spot, and from these evidences the DNA is extracted. Okay, now DNA is extracted. And this DNA by using restriction endonucleases. These are enzymes which will digest the DNA. So restriction endonucleases are the enzymes which will digest the DNA, that is, which are made into small fragments. So here you can see DNA is a long one thread-like structure which is made into the genomic DNA is made into fragments, that is genomic fragments. And these genomic fragments are subjected to gel electrophoresis. In this gel electrophoresis, they are separated according to the size. According to size, they are separated. And they are not next transferred to nitrocellulose membrane. That is blotting. That is by blotting. From gel, the DNA fragments are transferred to nitrocellulose plate. And next step is hybridization with the help of probes. So probes are single-stranded DNA fragments which are complementary to VNTR. So when they are left, we can see the bands like this. So by using X-rays, when you take autoradiography, where we take, by using X-rays, we take the autoradiography. So where we will get in the form of Philips. That is, the final output will be like this, where it shows banding pattern, that is nothing but VNTR. Okay. Here you can see the process, the entire process here. Okay. If you take the entire process, that is taking of DNA sample, okay, so extracting, that is nothing but extraction of DNA. That is, to extract DNA, we use many enzymes like proteases, like this. Proteases, okay, cellulose enzyme like this. Okay. After extracting, we'll get the genomic DNA, which is long thread-like. That is the genomic DNA which is long thread-like. So by using restriction endonuclease enzyme, which are called molecular scissors. So which are called molecular scissors. So by this we got the DNA fragments. So here we can see the fragments of DNA. And these fragments of DNA are separated by electrophoresis. That's separated by gel electrophoresis, where according to their size they are separated, that is short DNA fragments, long DNA fragments. By Blotting technique. It is transferred to nitrocellulose membrane, and where they are hybridized with the help of probes. That is, here hybridization is done. Hybridization means where the probes which are radioactive one, which are complementary to the VNTRs, they are. So by using X-rays, they go for autoradiography. So by the end, we'll get a output like this. There is a DNA fingerprint, which indirectly represents that is, so which will indirectly represent that is the VNTR. So here we'll see a case study. So what is this case study? So let us take a short DNA sequence like GC or AGCT, where repeated for number of times. So which is called VNTR. So which is called VNTR. And this GC sequence is present on, that is, if you take locus A, which is repeated for many times. Okay, so here you can see GC, 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 which is repeated in allele A2, A5, allele A3, A4. Okay. For example, if you take locus B, AGCT, which is repeated for many times, that is B2, B3. Okay, we can see locus B. So like this, we can see there are repeated sequences. Allele A2, A5, B2, B3, A3, A4. Okay, these are alleles. A2, A3, B3, B2, B4. Okay, like this. So this is the DNA fingerprint of individual 1 and this is individual 2. So when you take the individual 1 and individual 2, the sequences, here there is no A5, here there is no A2, no B3 here, only B2 here. So this is how the DNA fingerprint of one individual is compared with other individual. So during any crimes, the DNA fingerprint is done for an individual on the suspected one. Suppose here if we take individual 1 and individual 2. Like this, if you take, that is, if you take the individual 1 and 2, for example, if you take the DNA fingerprint at a crime spot. So this crime spot DNA which is collected in the fingerprint is compared with the suspects or who are already criminals. When it matches with this, he is considered as a crime. Like this, DNA fingerprinting is done. So what are the applications? The applications are, they are used in forensic science, that is to identify criminals. To establish paternity issues, that is family issues. And to trace evolutionary relationships, that is to trace the evolution of humans, in what way they are closely related to other animals. Okay. So not only in crimes, not only in forensic science in relating to paternity issues, in family relationship disputes, and to find out the evolutionary relationship. Okay. So till now we discussed about the regulation of gene, human genome project, and next we discuss about DNA fingerprinting. I hope you all understood. Follow us on SPS University in social media. Subscribe our videos. Thank you all. We'll meet in the next session.